In glycolysis and cellular respiration, a cell breaks down a molecule of glucose and uses its energy to form 32 molecules of ATP, an important cellular energy source. Most of this ATP is generated in mitochondria during the final phase of cellular respiration, during which electron transport and ATP synthesis take place. A mitochondrion has two membranes, an inner and an outer membrane, that divide the organelle into separate compartments. The inner membrane contains the components required for electron transport, called the respiratory chain, and ATP synthesis. The intermembrane space comes to be filled with protons pumped from the matrix by the action of the respiratory chain. NADH, along with FADH2, which is not shown, fuels the respiratory chain. NADH molecules carry high-energy electrons that were removed from glucose during glycolysis and the citric acid cycle. The respiratory chain consists of a series of electron carriers, each of which holds donated electrons tighter and with greater affinity than the previous carrier in the chain. When NADH donates electrons to the first carrier, NADH converts back to NAD+. The respiratory chain is important in part because it regenerates NAD+, which is required in glycolysis and the citric acid cycle. The electron transfer to the first carrier, called NADHQ reductase, drives the protein complex to pump protons across the membrane. NADHQ reductase then passes the electrons to a mobile molecule called ubiquinone. In turn, ubiquinone passes the electrons to cytochrome C reductase, a transfer that is coupled to the pumping of protons into the intermembrane space. Cytochrome C reductase derives its name from a redox reaction that it participates in. In redox reactions, molecules that gain electrons are said to be reduced, while those that lose electrons are said to be oxidized. Cytochrome C reductase reduces the next carrier, cytochrome C, by donating electrons to it. The next carrier, called cytochrome C oxidase, oxidizes cytochrome C by taking electrons from it. During the electron transfer, cytochrome C oxidase pumps protons across the membrane. The electrons occupy a relatively low energy state in cytochrome C oxidase, but can occupy an even lower state by joining oxygen and protons to create water. Oxygen is electron-greedy and serves as the final resting place for the electrons originally donated by NADH. Without oxygen, the complexes in the electron transport chain would be stuck with their extra electrons, and the chain would be blocked, unable to transport electrons from new NADH molecules. The process of transferring electrons along the chain releases a great deal of energy, which has been used to pump protons into the intermembrane space. This active transport of protons sets up an imbalance of both concentration and charge across the membrane. The imbalance represents potential energy. The protons have a tendency to flow down their concentration and charge gradients. However, they can only flow by passing through a channel in a protein complex called ATP synthase. As protons flow through this channel, they drive the ATP synthase to force together ADP and inorganic phosphate. ADP and inorganic phosphate are both negatively charged, and it takes energy, such as that embodied in the proton flow, to bring them together into a single molecule of ATP.